Synapses in many parts of the brain are not hardwired and unchangeable. Rather, they are plastic, in that their strengths can be adjusted by the degree of activity at the synapse. In this movie, we will discuss how high rates of activity can strengthen synapses through a phenomenon called long-term potentiation, or LTP. So here we're going to demonstrate the phenomenon of long-term potentiation, also abbreviated as LTP for short. An LTP is the strengthening of one synapse due to high rates of activity at that synapse. And the way we're going to demonstrate it is we're going to record postsynaptic potentials here in this little purple cell, and then we're going to attach a stimulating electrode to cell 1, the red cell, and a stimulating electrode to the axon of cell 2, indicated as pathway 2. And the first thing we're going to do is we're going to stimulate pathway 1, the red cell, with a single electrical shock and record the postsynaptic potential in the purple cell. Here we go. We stimulate it, generates an action potential, generates a postsynaptic potential in the purple cell, and that's what we record from stimulation of pathway 1. And then we're going to do the same thing in pathway 2. We're going to stimulate it with a single electrical shock and record the postsynaptic potential evoked by pathway 2, and it's exactly the same thing. Next, what we're going to do is stimulate pathway 1 titanically with a very, very high rate of action potentials that lasts for a few minutes. Watch. We stimulate it, very, very high rate, and then we stop. And now what we're going to do is stimulate pathway 1 again with a single electrical shock. And when we do that, what we see is the postsynaptic potential has increased markedly. It's more than doubled in amplitude. But if we stimulate pathway 2, the green cell, with a single electrical shock, nothing's happened. It's exactly the same postsynaptic potential as was evoked previously. In other words, there was specific strengthening here. Only synapse 1 was strengthened because only synapse 1 was titanically stimulated. Okay, so now what we can do is plot the complete record over time, and we're going to do that in the bottom panel. And what we're going to do first is we're going to stimulate cell 1 and cell 2, but very, very slowly. Boom, 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 alternate between the two, and we're going to get these control responses. The control responses are shown as the black records in pathway 1 and the black record in pathway 2. Then what we do is we're going to tetanize pathway 1. We're going to stimulate the heck out of it. And then what we're going to do is stimulate with single pulses, boom, 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 and record the responses. And you can see them that they're very, very large. These are the responses shown in red in pathway 1 after tetanus. They're much, much larger than the control. But if we do the same thing now to the green pathway, just stimulate it periodically, boom, 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 once every few seconds, there's absolutely no change. And the difference in the amplitude in the PSPs between pathway 1 and pathway 2 defines the amount of long-term potentiation due to the tetanization of pathway 1. The strengthening of the synapse that cell 1 makes with its postsynaptic target is due to the presence of NMDA receptors. As shown in the figure in the lower left, the synapses made by cell 1 and cell 2 have both AMPA and NMDA receptors. The critical feature is that the strong tetanic stimulation of cell 1 opened both the AMPA and the NMDA receptors at the synapse, which in turn allowed for a large influx of calcium through the NMDA receptors. The calcium in turn triggered a set of events which then strengthened the synapse in ways that are described in this and in the next movie. Before discussing how calcium can cause the synaptic strengthening, let's first review the cooperative action of AMPA and NMDA receptors. Recall that in order for ions to pass through the channel of the NMDA receptor, two events are necessary. First, the binding of glutamate has to open the gate on the channel, and second, the membrane has to be depolarized, which then expels the magnesium ion that normally plugs the pore of the channel. Here's how they work together. First, glutamate binds onto both channels and opens their gates. The opening of the AMPA receptor allows for the influx of sodium, which then depolarizes the membrane. If the depolarization is sufficiently large, the depolarization expels the magnesium ion, plugging the pore of the NMDA receptor, which is now open. The pore of the NMDA receptor is permeable to both calcium and sodium, and there is then an influx of both ions into the cell. The influx of calcium has major consequences because it ultimately strengthens the synapse if the influx of calcium is high, or weakens the synapse if the influx of calcium is low. The reason that calcium influences synapses is due to the action of a calcium-binding protein that occurs in all cells and the action of a protein kinase that is especially concentrated at the synapse. The calcium-binding protein is called calmodulin. When calcium enters the cell through NMDA receptors, it binds to and activates the calmodulin. The activated calcium calmodulin complex, in turn, then activates the protein kinase. The activated protein kinase can then phosphorylate other proteins, which include ion channels such as AMPA receptors, as well as a number of other proteins. There are a number of inactive kinases in neurons, but the kinase that is activated by the calcium calmodulin complex has a long name. It is called calcium calmodulin dependent protein kinase 2, or CAM kinase 2 for short. The details of how the CAM kinase 2 is activated will be explained in another movie that deals exclusively with the structure and operation of CAM kinase 2. But the important point at the moment is that the kinase is normally inactive, but once it is activated by the calcium calmodulin complex, it phosphorylates in other proteins and initiates a complex series of events. But now let's consider what the activated kinase actually does. First, there has to be a large influx of calcium due to the strong stimulation of the synapse. There, it's a large calcium signal. The calcium binds to the calmodulin and activates the CAM kinase 2. The activated kinase then phosphorylates a substrate, which then activates a series of very poorly understood events. But the net result of those events is that additional AMPA receptors are inserted into the synapse. Those new AMPA receptors can bind glutamate. So when the synapse is activated again, there are now more AMPA receptors that are opened. And hence, there is more sodium influx into the cell, and thus a larger depolarization than there was before the new AMPA receptors were inserted. But the activated CAM kinase 2 has a second effect. It directly phosphorylates the AMPA receptors at the synapse. The phosphorylation increases the channel conductance. That is, it allows more current to flow through the channels when the channel opens. Thus, the total effect of the CAM kinase 2 activation is both to insert more AMPA receptors into the synapse and to increase the amount of positive charges that can flow through each AMPA channel due to the phosphorylation of each of the receptors. Thus, the strong stimulation of the synapse causes a large calcium signal that potentiates or strengthens the synapse, where the release of glutamate from the presynaptic cell now generates a much larger postsynaptic response than before the strong signal was presented, due to both the increased number of AMPA receptors and their enhanced conductance. Since these effects last for an hour or more, the phenomenon of synaptic strengthening is called long-term potentiation. Here we saw how a synapse can be strengthened by high rates of activity, which causes strong synaptic stimulation. In the next movie, we are going to see the exact inverse of these effects, specifically how low rates of synaptic activity can weaken a synapse, a phenomenon called long-term depression. Indeed, the synaptic weakening with low rates of stimulation can actually reverse the events of the synapse produced by long-term potentiation.